Let's talk about DNA replication. DNA replication is divided into three basic phases. Initiation starts at a specific nucleotide sequence known as the origin of replication. Here, several inhibitor proteins bind to the DNA and begin the process of unwinding with the help of the helicase enzymes. The helicase enzymes break the hydrogen bonds that link the complementary base pairs of the nucleotides. There are also single-strand binding proteins that help to stabilize the newly unwound single strands. These single strands act as a template that will give a guide when a new polypeptide is being synthesized. So in summary, during the DNA replication initiation phase, the DNA strands unwind, resulting to the formation of a replication bubble. The replication bubble leads to the formation of replication fork, so the DNA replication will begin outwards from the two replication forks that were formed during the unwinding of the DNA strands. DNA replication will then continue in both directions, resulting to a replication fork at the end and formation of a new polypeptide.
Elongation synthesizes new DNA strands by joining individual nucleotides together. DNA polymerase III is the enzyme that catalyzes the addition of new nucleotides only to the 3' hydroxyl free end of the nucleotide. Also, we must take note that DNA polymerase III can only synthesize new DNA in the 5' to 3' direction. In this process, there are two types of strands. The first strand is the leading strand and the second strand is the lagging strand. In the leading strand, the DNA polymerase III follows its normal reaction route. That means it adds nucleotides to the 3' free end of the chain of nucleotides, and this is always in the direction of the replication fork. However, in the lagging strand, the DNA polymerase adds new nucleotides to the 3' hydroxyl end of the RNA primer, which is a short segment of RNA that is complementary to the 3' to 5' part of the DNA template strand. And this usually serves as a starting point for the addition of nucleotides. This movement is usually away from the replication fork. After the addition of each primer, the synthesis of the Okazaki fragments, which are a series of short fragments of DNA that each begin with a section of RNA, takes place. Then comes in enzyme DNA polymerase I, which is responsible for removing the RNA primers and filling the spaces by extending the neighboring DNA fragments. The Okazaki fragments are then joined by an enzyme known as DNA ligase.
In the termination phase, the replication process is completed, and the two new DNA molecules separate from each other. And as soon as the new formed strands are complete, they rewind back automatically into their helical structure. The protein DNA complex that carries out the replication fork is often referred to as the replication machine. Many key enzymes are involved. For example, helicase helps to unwind the parent DNA, and primase synthesizes RNA primer that is used to generate Okazaki fragments. As for topoisomerase II, it helps to relieve the strain on the structure of the parent DNA that is generated from the unwinding of the double helix.